Hey guys, my name is Shrek Harigaonkar, and today we're going to talk about cancer genes and gene fusion in cancer. So, when we talk about any question in the exam, one thing you need to do is what I would suggest is create like a map of what you want to talk about and jot down like specific important points. So, in this question, the important points would be cancer genes and different types of genes and genetic lesions in cancer, and that you would have like what is it about the driver and passenger mutations and epigenetic modifications in cancer. So let's get right to it. So whenever you start talking about any question, you have to talk about the you want to start with the definition. So in this case, it would be defined as genes that are currently affected by genetic changes in cancer, presumably because they contribute directly to the malignant behavior of cancer cells. So, in general, like you can have different kinds of mutations that may give different kinds of mutations in cancer genes, which can affect its function, and as a result, you have development of a precursor or direct cancer. So, if such mutations drive carcinogenesis, each cell in an individual tumor shares mutations that were present in the founding cell at the time of malignant transformation. So let's start with the different types of genes. So the first gene is oncogenes. So these are basically mutated proto-oncogenes, and proto-oncogenes basically promote cell division. When they're mutated, you may have uncontrolled and unregulated cell division, which gives rise to cancer. And these proto-oncogenes are then known as oncogenes. So let's see what we wrote. So induce a transformed phenotype when expressed in cells by promoting increased cell growth. Yeah. So you have different categories within oncogenes as well. So these may involve growth factors, for instance, PDGF, which is platelet-derived growth factor, or VEGF, which is vascular endothelial growth factor. You may also have growth factor receptors like HER2 or NEW in breast cancer, and signal transducers, for example, RAS, which is common in colorectal carcinoma, for instance. You can also have cell cycle regulators, for instance, in cyclin D1 in mantle cell lymphoma, as well as CDK4 in melanoma. So oncogenes are considered dominant genes, and this is mainly because um, when there's a mutation in a single allele, it's usually sufficient to produce a pro-oncogenic effect. The next one is tumor suppressor genes. So these are basically genes that normally prevent uncontrolled growth, and when mutated, they allow the transformed phenotype to develop. These are known as recessive genes, and that's because both copies of the gene must be knocked out for tumor formation. So within this itself, you have two groups, and one of them is the gatekeepers or governors. So these basically they regulate transition of cells through checkpoint in the cell cycle, and these involve retinoblastoma protein, the CDK inhibitor like P21, P27, P53. You also have caretakers or guardians, which are responsible for protecting the integrity of the genome by encoding certain proteins that are involved in detecting and repairing mutations. So some of these examples, like as mentioned, P53 and retinoblastoma genes of tumor suppressor genes, these are usually the most classic examples. Uh, P53 usually regulates the progression of the cell cycle from G1 to S phase. And in response to DNA damage, P53 also slows the cell cycle and upregulates DNA repair enzymes. So if DNA repair is not possible, then P53 gene, the gene protein, may induce apoptosis by upregulating BAX, which is basically BAX, and that disrupts BCL2, which eventually leads to cytochrome C leakage from the mitochondria, which results in apoptosis. So BCL2 is another, it's involved it's like a oncogene basically. Yeah. So then you have genes regulating apoptosis. So these basically prevent apoptosis in normal cells, but promote apoptosis in mutated cells whose DNA cannot be repaired. So you have some examples over here. As mentioned, you have BCL2, which normally stabilizes the mitochondrial membrane and blocks release of cytochrome C. Disruption of BCL2 allows cytochrome C to leave the mitochondria and activate apoptosis. So BCL2 is overexpressed in follicular lymphoma. So in follicular lymphoma, it's basically, as mentioned, is the translocation in 14 and 18. So you usually have the BCL2 gene, which moves from chromosome 18 to chromosome 14. And in chromosome 14, you usually have the immunoglobulin heavy chain locus. As a result, you have overexpression of BCL2, and this helps prevent apoptosis. And if you don't have apoptosis, you have accumulation of different kinds of cells or a large amount of cells of a specific type, and hence you have cancer, and in this case, you have lymphoma. You also have genes regulating interaction between tumor cells and host cells. This one is kind of low yield, but it's important to mention. So these genes are also recurrently mutated or functionally altered in certain cancers, and particularly important genes are genes that enhance or inhibit recognition of tumor cells by the host immune system. Then you have genetic lesions in cancer. So what is it? So the genetic changes found in cancer vary from point mutations, and there are different types of genetic lesions, which we shall cover in the upcoming slides. In certain neoplasms, genetic abnormalities are usually non-random and highly characteristic. So whenever we talk about genetic lesions, we need to talk about what driver and passenger mutations are. So driver mutations are basically mutations that alter the function of cancer genes and thereby directly contribute to the development of progression of a given cancer. So these are usually acquired, but occasionally inherited as well. Passenger mutations are acquired mutations that are neutral in terms of fitness and do not affect cellular behavior. So they just, like you can say, they just come along for the ride. For example, we'll come to examples over here. So despite their apparent innocuous nature, passenger mutations have nevertheless proven to be important several ways. So in carcinogen-associated cancers, a mutation analysis have provided evidence that more genomic damage is directly caused by the carcinogen. So one example of this, for instance, would be like before the sequencing of melanoma genes, the causative role of sun exposure in this cancer was debated. So nowadays, it's we know that melanomas have thousands of mutations of a type that is specifically linked to the damage caused by ultraviolet light. Um, a second, more nefarious effect of passenger mutations is that they create genetic variants that, while initially neutral, they may provide tumor cells with a selective advantage in the setting of therapy. So this is basically associated with the development of resistance to chemotherapy in treatment of cancers. Coming to the point mutation, which is the type of genetic lesion. So as we know, a point mutation occurs in a genome when a single base pair is added, deleted, or changed. So these may involve like silent mutations, nonsense mutations, missense mutations, and frame shift mutations, which I'm sure you're aware of in genetics. So mutations can either activate or inactivate the protein and products of the affected genes, depending on their precise position and consequence. So in general, you have like different kinds of genes. So one cardinal example is mutations that, for instance, convert the RAS gene into a cancer gene, which is one of the most common events in human cancers. So you can have point mutations in oncogenes, as mentioned. As a result, you have increased growth. Or in contrast, you can also have point mutations in genes that is associated with a loss of function, characteristically TP53, and it's known that in approximately 50% of cancers, there's a TP53 mutation. You also have gene rearrangement, which is pretty important. So these may be produced by chromosomal translocations or inversions, and these rearrangements can activate proto-oncogenes in two ways. So some gene rearrangements result in overexpression of proto-oncogenes by removing them from their normal regulatory elements. So these are highly seen in lymphomas, non-Hodgkin's lymphomas like Burkitt lymphoma and follicular lymphoma, as we mentioned. Uh, in more than 90% of cases of Burkitt lymphoma, they usually have a translocation, and that's usually between 8 to 14, 8, 14. So this basically leads to the overexpression of the MYC oncogene on chromosome 8 by juxtaposition with the immunoglobulin heavy chain gene regulatory element on chromosome 14. And you also have follicular lymphoma, which is a reciprocal translocation between 14 and 18, which leads to overexpression of the anti-apoptotic gene, BCL2, as mentioned before. Um, and you have Philadelphia chromosome, which is very common in CML, which is often a characteristic feature of it. And this is a very important example as well. Then you have deletions. So deletions are another prevalent abnormality in tumor cells. So as mentioned, you can have deletions in tumor suppressor genes, which is pretty common, which leads to inactivation of both alleles. Usually, they require both allele inactivation in order for them to contribute to carcinogenesis. So some examples, for instance, may be deletions involving 13q14, which is the site of the retinoblastoma gene, and that's associated with retinoblastoma. And you also have deletion of the 17p, which is associated with loss of TP53. The next type of genetic lesion is gene amplification. So in this, proto-oncogenes may be converted to oncogenes. So if you think about it, it just makes sense. If an proto-oncogene is highly amplified, it turns into an oncogene because of increased uh, proliferation. Two clinically important examples of amplification are NMEC, the gene in neuroblastoma, as well as the HER2 gene in breast cancers. Then you have aneuploidy and microRNA. So aneuploidy, as you know, is defined as a number of chromosomes that is not a multiple of the haploid state. For humans, the chromosome number is usually not a multiple of 23. So usually it's 23, but in aneuploidy, it's not a multiple of 23. So in this, simple examples would be aneuploidy of portions of chromosome 17, where the TP53 gene is located and often lost. As a result, you have development of malignancies. The next one is microRNAs. So these are basically non-coding single-stranded RNAs, and microRNAs, in general, they inhibit gene expression post-transcriptionally by repressing translation, or in some cases by messenger RNA cleavage. So they may have a role in both uh, developing of the cancer as well as suppressing of cancers. So, for instance, there's an example over here. If the target of the miRNA is a tumor suppressor gene, then it suppresses the tumor suppressor gene. As a result, you have cancers. On the other hand, if it suppresses an oncogene, and that if the mRNA suppresses an oncogene, and that mRNA is there's a mutation with it, then it'll be associated with high levels of expression of the oncogene. As a result, you'll have malignancies. And finally, last but not least, you have epigenetic modification than cancer. So epigenetic modification basically refers to changes in the gene expression that are not caused by changes in the DNA sequences, but are due to events like DNA methylation, histone modification, etc. So in normal differentiated cells, 80% of the genome is not expressed. These regions of the genome are usually like silenced by DNA methylation and histone modifications. On the other hand, cancer cells are characterized by a global DNA hypomethylation and selective promoter localized hypermethylation. And this is an example involving the Notch1 gene, which has an oncogenic role in T cell leukemia, especially. So yet acts as a tumor suppressor in squamous cell carcinoma. So in different kinds of cells, it might act differently, and that is basically what epigenetic modifications involve. So as a result, as mentioned, there's a dichotomy that exists between the squamous cell carcinoma, the expression of Notch1 in squamous cell carcinomas, and in T cell leukemia specifically. So that's all from me. Thank you.